Today is part 3 of my discussion on amino acids. Here are your 20 stinking proteogenic amino acids. You need to know their names, the 1-letter abbreviation, and the 3-letter abbreviation. Proteins, when you digest them and break them down, they become polypeptides, and then oligopeptides, and then dipeptides, and then amino acids. Let's start by answering the question of the previous video. All of the following are sulfur-containing amino acids except, and the answer is valine. Cysteine has sulfur, methionine has sulfur, homocysteine has sulfur. If cysteine has sulfur, then homocysteine does. But methionine, how come? Remember, methionine will give you SAM, and SAM is the ultimate methyl group donor. The way I remember it is that methionine has methyl, methionine has sulfur. You remember that? That's what we're talking about. Methionine has methyl, methionine has sulfur. That's why it can give you SAM, methyl, it can give you cysteine, sulfur. The amino acid has an amino group, a carboxyl group, and an R group side chain. Amino group on the left, carboxyl on the right, A before C. Who determines the chemical properties? The R. We talked about their classification in the previous video. First group, nonpolar, non-aromatic side chain, and these include valine, leucine, isoleucine. And we talked about maple syrup urine disease in the last video. Methionine has methyl, methionine has sulfur. Proline is like a pentagon, it is cyclic. And then we talked about the Gordon-Ramsay amino acids. They are so aromatic. FME, phenylalanine, tyrosine, and tryptophan. Next, we have the polar. Some of them contain OH, serine, and threonine. Some of them contain sulfur or thiol, cysteine. Some of them are amides, asparagine, glutamine. The negatively charged, or the acidic ones, aspartic acid, glutamic acid. When you deprotonate aspartic acid, it gives you aspartate. When you deprotonate glutamic acid, you get glutamate. These are negatively charged. These are acidic. And then you have the positively charged or the basic, P or B. You have the arginine, lysine, histidine. Hormones, or generally speaking, any molecule could be lipid-soluble or water-soluble. Lipid-soluble is the same as fat-soluble, water-insoluble, non-polar. Water-soluble is lipid-insoluble, but polar. And in biology, you learn that your cell membrane is made of fat. It's a lipid bilayer. It is fatty. But the blood, the plasma, is watery. Therefore, if this protein is gonna float in the plasma, you gotta put the water-soluble stuff on the outside because it is coming in contact with the water that's in the plasma. Makes sense. How about the lipid-soluble portion? Put it in the center, in the interior. If you have an alkyl side chain, you will be strongly hydrophobic. What the flip is hydrophobic? Hydro, water. Phobic, afraid. Oh, I'm lipid-soluble. Water, insoluble. Put me in the center, baby. Examples, the alkyl side chains, valine, leucine, isoleucine, alanine, and don't forget phenylalanine. The first 4, nonpolar, non-aromatic. The last one is aromatic. It smells. Think of Gordon Ramsay saying, oh, F me, it's aromatic. Positively charged amino acids, such as what? Arginine, lysine, histidine. And negatively charged amino acids, such as what? Aspartate and glutamate. Because remember, aspartic acid, glutamic acid, these are acidic, negatively charged. How about the amide? Asparagine, glutamine. Here's a very good rule of thumb. If you are charged, like positive or negative, you are hydrophilic. Absolutely. Because you are polar. You're polarized into positive or negative. Therefore, I'll put you on the periphery. So all of the amino acids here are hydrophilic. Next, amino acids are amphoteric. What the flip is that? You know an amphibian? What's an amphibian? Oh, an amphibian is an organism that can live in sea and on land. Thank you so much. So the word amphi means double. Amphibian. Amphi, double. Bian from bio, which means life. Here's amphoteric. Ampho means what? Ampho means double. I live a double life. Basic portion, acidic portion. Amino acids are amphoteric. They have a basic part, acidic part. They can accept a proton, they can donate a proton. Who will decide? The pH of the medium will decide. Are you in an acidic environment or in a basic environment? In an acidic environment, the ionizable group will gain a proton because the acid has too much proton to give. All right. How about in a basic solution? Basic solution doesn't have any protons. All right. So you will lose protons and give it to the solution in order to try to achieve equilibrium. This is how you create order from chaos. So when the pH of the medium is low, ionizable groups are becoming more pronated. They are gaining a proton. But in a basic solution with high pH, the ionizable groups are becoming depronated. They are losing protons. In his famous book, Charles Dickens said, they can accept protons, they can donate protons. He was talking about amino acids and their amphoteric properties. If you remember my videos on pharmacokinetics and pharmacodynamics, I've told you that a singular drug can have non-ionized portion and ionized portion at the same freaking time. What determines the degree of ionization? The pKa of the drug and the pH of the medium in which the drug is present. What if pK and pH are equal? At that point, you will have 50% of the drug as non-ionized and 50% of the drug as ionized. So what is the pK? It's the pH at which the non-ionized portion equals the ionized portion. In other words, the pK is the pH at which the protonated fraction equals the deprotonated fraction, 50-50. So when the pK equals the pH, the protonated equals the deprotonated. Hashtag equality. To understand the difference between pH and pK, imagine that medicosis is swimming in the water. Okay, medicosis is the pK. The water has a pH. All right, the water can get colder or warmer. It can get more acidic or basic, but medicosis does not change. Same thing, pK is a freaking constant for a singular molecule. The pH is the pH of the medium. You can add some acid to the medium to make it more acidic. In this case, the pH will go down. Or you can add more bases to the medium to make it more basic. In this case, pH will go up. But the pKa is the pKa. Medicosis is medicosis. It's a constant. So there are 3 rules. If you understand them, you will answer any question correctly. Rule number 1, pK is the pH at which half is protonated and half is deprotonated and they equal each other. But what if the pK is greater than the pH? Imagine that the amino acid exists in an acidic environment because an acidic environment will have a very low pH to the point that pK exceeds the pH. All right, you put me in an acidic environment, I'll give you more protons. Yep, the protonated fraction will exceed the deprotonated fraction because this is a freaking acidic medium compared to the pKa. Conversely, if the pKa is lower than the pH, then the protonated form is lower than the deprotonated form. If you want to make it easy on you, always start with the pKa, all right? And always start with the protonated. So pK protonated. Nice. And then when this is greater than, this is greater than. When this is less than, this is going to be less than. When this is equal, this is equal. Duh. An amino acid has an amino group and a carboxyl group. Each one has a separate pKa because this is Peter, this is Paul. Since an amino acid has 2 groups that can be protonated, the same amino acid will have 2 pKa's. What if the amino acid has 3 groups? Then it will have 3 pKa's. So let's say that this is carboxyl group and it has a pKa 1 of about 2. How about this one? Has another pKa of about 10. They are not the same. What if you put this amino acid in a medium that has a pH of 2? Well, at this point, half of the carboxyl group will be protonated, the other half will be deprotonated. So let's do this. First thing, you write the 3 equations and let's go. At a pH of 1, what's going to happen? Look at the first pKa, about 2. The second pKa, about 10. All right. Is the pKa greater than pH? Here, yeah, 2 is greater than 1 and 10 is greater than 1. Therefore, both of them, carboxyl and amino acid, will be what? In the protonated. They will be fully protonated. What does that mean? Protonated means proton, H. Therefore, you will see it as COOH and not as COO. And you will see this as NH3 plus rather than NH2 because this has more protons. And at this moment in time, you have NH3 on one side and COOH on the other one. Okay, this one has no charge. This one has a positive charge. Therefore, the entire amino acid is positive. Thank you so much. Let's change the pH. Now the pH is 6. Let's talk about Mr. COOH. All right, is the pK greater than the pH? 2 is not greater than 6. Therefore, we follow rule number 3. When the pK is lower than pH, the deprotonated will exceed the protonated. Oh, look at this. Most of this is COO negative, deprotonated baby. How about the other doofus, the amino group? Look at it. The pK is 10. Is this greater or lower than the pH of 6? This is greater. Therefore, more protonated, please. I exist in the NH3+. Therefore, my carboxyl exists in the COO negative, but my amino exists in the NH3 positive. A positive wing and a negative wing. That's why this entire amino acid is neutral. It is dipolar. It has a positive pole and a negative pole. We call this a Zwitter ion. So the Zwitter ion in German means hybrid. So if you're driving a Toyota Prius in Germany, they call it Zwitter ion. I'm just joking. But what if the pH of the solution is about 7.4, which is the normal pH in your arterial blood? Now pause and review. It's the same answer as when the pH is 6 because this lies in between 2 and 10. It's again a Zwitter ion. Now, what if the pH is freaking 11? Oh, 11. All right, let's start with Mr. Carboxyl. All right, is 2 greater than or lower than 11? Of course, 2 is less than 11. Therefore, I exist in the deprotonated. Thank you so much. How about the amino side? 10 is lower than 11. Oh, also deprotonated. So I exist in NH2 rather than NH3. Look at my 2 wings. The first wing has a negative. The second wing is neutral. So what's the entire amino acid? The entire amino acid is negative. Now, let's talk about the titration of the amino acid. Before you freak out, oh my goodness, shut up, it's easy. What does titration mean? Gradual change. Gradual change. That's it. Look at this beautiful graph. You have pH here, pH of the medium. Is it more acidic or more basic? And here is OH, not HOH. As you know, OH is basic. So as you go from here to here, we're adding more bases to the solution. And therefore, what's going to happen to the pH as you add more bases? It's going to go up. And that's why we're going upwards in this direction. As you increase the bases, pH is going to rise. I'll see you in my next video. I'll make you master the curves of titration like a pro. But for now, please memorize these 3 equations. Some pearls for the pros. What's the difference between amino and amine or amino? Okay, amino is C bound to N with a single bond. But look at amino, double bond. Don't forget the pH and the pKa. They are also involved in something else called the Henderson-Hasselbalch equation. What if the numerator equaled the denominator? Now, when this equals this, the ratio is going to be 1. What's the log 1? 0, baby. And now the pH equals the pK. Of course, as you add more bicarbonate, pH goes up, and as you add more carbon dioxide, pH goes down. If you want to learn more about the pH, check out my acid-base imbalance course on my website, medicosisperfectionalis.com. Thank you for watching. Please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to my website to download my premium courses. Thank you for watching. As always, be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine makes perfect sense.